Good morning, and today is Mitochondria Day. We learn about how the mitochondria can act in our bodies and be related to aging. First, we had David Sinclair, and you know he gave a supplement, and by giving that supplement, which I talk about in other videos, he reversed the age of his mice and made their muscles stronger. But what if we went at it through the opposite direction? What if we tried to make the mice older? Well, and then, by the mechanism that we use to make the mice older, turned it back off and turned them young again. Now, doesn't that sound like science fiction? But it's actually true. There are two groups of people, one at the University of Alabama at Birmingham, and the other at the Birmingham Veterans Affairs Medical Center at Birmingham. And what they did is they took a mouse and they engineered the DNA polymerase in it, so it was basically a knockout mouse, well, almost a knockout mouse, so they could turn this DNA polymerase on and off. And this allowed them to basically shut down or slow down the function of the mitochondria a great deal. And by doing so, they were able to see how the mitochondria acts in controlling aging in mice. And they basically, for one thing they looked at, was skin wrinkling. So, let's just do a very quick review for those of you to remind you what organelles are, mitochondria, and so forth. Now, in the cell, like the body, we have the heart, we have the lungs, and so forth. We have tiny specialized structure within the cell, and these structures within the cell each have a specialized function. Take a look at this. I made a couple little videos for you to take a look at so you can follow along. So, the mitochondria is an organelle within the cell, but we can start off with the nucleus. The nucleus contains DNA. There is the endoplasmic reticulum, where ribosomes make proteins from RNA. There are the Golgi bodies, which temporarily store proteins in the cell. We have lysosomes that store digestive enzymes. And it also has vacuoles, which are small membrane-bound vesicles that can store water or food. And finally, our mitochondria, which via the electron transport system of the cell makes ATP, the energy currency of the cell. Now, let's take a switch right here and let's dive a little bit deeper and let's look at the mitochondria. Now, here we have a mitochondria. The mitochondria is a small double membrane structure believed to have once been a bacterium. And it became a symbiote within our cells because the cells at that time couldn't use oxygen, and the mitochondrial bacteria could. And now they became a symbiote and eventually merged to live within our cells permanently. They have their own DNA that's read by a slightly different code than the main DNA in the cell's nucleus. And located within the inner membrane of this double membrane structure is the electron transport chain, where ATP is formed. Okay, back to the mice. So they were actually able to induce hair loss and wrinkles in these mice and turn it back off again. So let's dive down this rabbit hole and take a look at some of the results. The mice, when DNA polymerase was suppressed, had reduced mitochondrial DNA content, reduced mitochondrial gene expression, and instability of supercomplexes involved in oxidative phosphorylation. Or in short, they produced a lot less energy. Well, they say a picture is worth 1,000 words, so let's take a look at this picture. First, take a look at mouse 1. Mouse 1 is the wild type, looking healthy and sassy. Mouse 2 is with the DNA polymerase suppressed for 2 months. They added a drug that turned off this DNA polymerase, but only in these mice specially sensitive to it. It doesn't look very pretty, does it? I think it looks, well, old. Now 3 is the kicker. 3 is the same mouse as 2, with the mitochondrial DNA polymerase allowed to express normally. 2 months later, this mouse has returned to a normal-looking age. The depleter mice, as they referred to them, when they had the reduced mitochondrial function, had inflamed wrinkles, alopecia, and hair loss. Now, if you look at a very old man, what do we see? Ah, you got it. So here's the title, Reversing Wrinkled Skin and Hair Loss in Mice by Restoring Mitochondrial Function. It's a very, very interesting article. There's a lot more meat into it. I really suggest that you do read it and dive into it. You can see from the one picture that I did put up that there was a lot of analysis of this data. And so I just wanted to kind of point you in the right direction with that. But I found that was one of the more fascinating papers I've recently read. So, on to part 2. Mitochondrial dysfunction or lowered activity is actually related to a number of those pesky diseases of aging, such as cardiovascular disease, cancer, even diabetes and some neurological diseases. So, another thing that comes up that's very important is, remember apoptosis, when the body recognizes dysfunctional or dead and dying cells and causes them basically to self-destruct. This mechanism is actually regulated by proteins within the mitochondria. So if the mitochondria isn't functioning properly, apoptosis isn't turned on and you get a buildup of these senescent cells, which then can cause inflammation and disease. So that's the connection. So we're going to now go to the sirtuin genes. Now remember the NAD that we talked about, and we had lowered NAD as we got older, and we're trying to turn on these sirtuin genes. Well, let's get into what's the nuts and bolts of this. So take a look at this chart. In mammals, there are a total of 7 sirtuin genes, sometimes called SIRT for short, and all are NAD dependent, and they're located in different parts of the cell. SIRT 1, 6, and 7 are the main nuclear ones, whereas SIRT 2 is found both in the nucleus and the cytoplasm, and SIRT 3, 4, and 5 are found in the mitochondria and responsible for protein acylation. Now, SIRT 3 appears to be the main regulator of mitochondrial metabolism and function. Now, there was a recent paper on this. I'll put the title up here. Come on, paper, paper, paper. Here, let's put it nice and big for you. A recent paper, Acylation of Mitochondrial Proteins in the Heart. The role of SIRT3 explores the role of modification of proteins in the cell by a process called acylation. Adding an acyl group to mitochondrial proteins has been shown to be involved in a number of diseases, including heart disease, diabetes, and even aging. So, adding and removing these acyl groups is turning on and off these genes that are involved. Now, removing the acyl group, which the acyl group being stuck on it is what's related to the disease, removing the acyl group is done by an NAD dependent, see the link? An NAD dependent deacylases, and they're produced by the sirtuin genes. So, I know some of you have asked about FOXO3 and a few of the other things, so let's take a look at this next chart and kind of dive into this. Okay, so adding and removing acyl groups is often involved in the energetics of the cell. So as you can see from this table, the transcription of DNA is upregulated when the protein products of things such as the FOXO3 gene is deacylated by a sirtuin gene deacylase. I hope you're starting to see how all this links together. This particular paper looked at the SIRT3 and how it was basically involved in cardiac disease. Okay, let's move on to, you've all heard of calorie restriction. We're still in the same paper here, but let's talk about that. I'm going to put another chart up here for you to look at. Calorie restriction also affects this enzyme. The acylation of these proteins are controlled by a rate-limiting enzyme, a phosphoribosal transferase, that makes the NAD in the cell. So calorie restriction affects the phosphoribosal transferase in a beneficial way. But a hack around this might be more effective is just to increase the NAD in the cell by an NAD precursor. So instead of starving yourself, and there's also some glitches that have come up with the monkey studies recently that showed that they lived longer, showing that, well, it wasn't really, the results weren't that great, but maybe I'll do another video on that at a later date. So as we age, proteins in our cells can get turned off by adding an acyl groups. The proteins can be turned on by removing an acyl group. The NAD-dependent sirtuin gene deacylases can turn them back on. The cells become more energetic and function better with more active players. The SIRT3 gene has a regulatory role where it causes a deacylation of FOXO3 and another gene called PARKIN to control autophagy and mitophagy. Mitophagy is basically apoptosis for mitochondria. And protein acylation in the mitochondria has been linked to a number of diseases of the heart, including hypertrophy, where the heart is abnormally enlarged, myocardial infarction, and heart failure. So the authors of this paper summarize in great depth a lot of what's happening with the sirtuin genes, and they basically conclude the SIRT3 activity controls the bioenergetics of the mitochondria. So that's very, very important. So one of the primary mechanisms to help control this, or to control the bioenergetics of the mitochondria, might be just to increase the NAD. So I think we know we've heard that refrain before. So there's a lot of interesting data in this paper, and I really suggest you, again, take a look at it. Take a look at the chart, and you can see, I'll put the chart back up here, you can see that, the NAD can have precursors such as diet. You can get it from tryptophan, from niacin, that takes you to NMN to NAD to NAD, or you can take NMN directly and so forth. And it shows you the chart here related to how it's associated with cardiac hypertrophy and heart failure in this particular paper. Okay, on to the next topic. Part 3. Okay, we all know that one of the things that we try to do is we eat a high antioxidant diet. We try to reduce the inflammation of our body because we know inflammation is vaguely somehow involved in aging. How exactly does this work? Well, in every cell we have a huge protein. It's known as the inflammasome. And the inflammasome actually is related to turning on inflammation. And what happens is the cell turns this on, and it can actually create something called pyroptosis, which is fire. The cell is so much inflamed, it's because it's on fire, and it can cause the cell to basically destruct. So let's take a look. Let me put a chart up here again. We're going to go up to the chart and the title of the paper and so forth. So first the title, and then I'll put the chart. So inflammation, when the body detects a pattern of the molecular patterns that tell the host, us, whether the molecules in our body are seeing related to a dangerous pathogen or a danger-associated molecule pattern such as that in uric acid, it starts to create inflammation. It basically activates the inflammasome, and it has all these inflammatory cytokines that are turned on. Which, if we're fighting off bacterial invaders or a virus, that's good. We want to basically send everything out there, have them release their free radicals, do whatever damage they can to kill the bacteria. But if we're reacting to something like, say, uric acid in a joint, that's not a good thing, because then you're not going to kill the uric acid crystal, and you're only damaging the tissue around it, like for instance, in the case of gout. So in the diagrams we see here, we see first the cascade is initiated via a raise in calcium ions. That triggers the activation of the inflammasome unit, NLRP3. That's a tongue twister. Say that 3 times fast. And this leads to the release of immune system signals in the form of inflammatory cytokines. The second diagram shows how reactive oxygen species, you may be more familiar with the term free radicals, in this case containing a molecule of oxygen, which activates the inflammasome. Now, these reactive oxygen species can be generated by a number of different mechanisms, but when the proteins that are involved in the autophagy of mitochondria are inhibited, the malfunctioning mitochondria are not disposed of, and we get more of them. And the more malfunctioning mitochondria we have more inflammation production, and this raises the total inflammation of the body, and is often linked to the triggering of disease. So we see inflammation in many of our diseases of aging, from arthritis to cardiac to neurological problems. A better understanding of the molecular basis of this, I believe, will lead to treatments and perhaps even prevention in the human body. So I hope you've enjoyed this deeper dive into the mitochondria and how it can be involved in aging. Because one reason when I started this channel was someone would see something in a paper, and they would see, like, for instance, calcium upregulating an inflammatory thing. And that's only when it's released in a specific area of the cell. It doesn't mean to stop taking your calcium supplements if you're taking it to prevent osteoporosis and so forth. So I would see people jump in and make some sort of very wild claim that changing one thing is going to radically change how your body reacts or cure you in some way. All of these things are much, much more involved and much, much more detailed. And turning on one thing might turn something else off. And sometimes you might need one thing on and one thing off, or vice versa. So these pathways need to be very, very well understood, and how they work in the human body, not just, say, in mice or worms or something like that, before we really start implementing them on ourselves. I really think some great things are coming. I'm hoping in the near future. I have been collecting some information on senescent cells and some of the things that help the body clear senescent cells, because there have been some very interesting trials at the Mayo Clinic lately. So I'm very much hoping I'm gathering the information, and hopefully I will have that out to you soon. And anyhow, I hope if you like this video that you will subscribe, and that you will give me a little like, hit the little like button wherever it is on your screen, whether you're on the TV or the computer or your phone. And until next time, this is Judy Chalice with Lifespan and Longevity. And remember to like and subscribe.